You're reviewing Daraxonrasib or chemotherapy in previously treated metastatic pancreatic cancer by O'Reilly M., Wainberg Z.A., Hendafar I. Borid M.J., Pietrantonio F. Pant S. Hamel P. Walpin B.M. Published in New England Journal of Medicine. Scientists called it the Death Star. Smooth, round, no pockets for a drug to grab onto, and it sits at the center of nearly every deadly pancreatic tumor on Earth. For 40 years, KRAS was the most famous cancer gene nobody could touch. Then, in a packed plenary hall in Chicago in 2026, that changed, and the room reportedly gave the data a standing ovation.
We're talking about Daraxonrasib and the RASolute 302 trial, which nearly doubled survival in previously treated metastatic pancreatic cancer. But to understand why oncologists jumped to their feet, you have to go all the way back to two sick lab mice in the 1960s. This is the full 40-year story: the virus hunters, the chemists who found a crack in an unbreakable protein, and the trial that just rewrote what's possible.
And if you want more evidence-first breakdowns like this, the real data, the real history, no hype, hit subscribe now, because we're about to get into exactly how this happened.
The KRAS story doesn't start in a hospital, it starts in a virology lab. In 1964, Jennifer Harvey isolated a rat sarcoma virus carrying a gene we now call HRAS. In 1967, Werner Kirsten found a sister virus carrying its cousin, KRAS. For years, these were just viral curiosities, until Edward Skolnick and colleagues showed the viruses had actually stolen these genes from normal animal cells in the first place.
Then came 1982. Researchers still call it the year of the Oncogene. Channing Dare, working in Jeffrey Cooper's lab, and separately Mariano Barbacid, Robert Weinberg, and Michael Wigler, took DNA straight from human tumors and showed the same genes could transform normal cells into cancer cells in a dish. The trigger was almost absurdly small. One single letter change in the gene, usually at codon 12, was enough to jam the switch permanently on.
The link to pancreatic cancer came in 1988, when Carmen Almaguera, Nehiko Shibata, and Manuel Paracho published the first hard evidence that KRAS mutations were driving human pancreatic tumors. Later genome sequencing projects confirmed just how dominant that signal is. Roughly 90 to 95% of pancreatic ductal adenocarcinomas carry a KRAS mutation, mostly at three hotspots: G12D, G12V, and G12R.
That created the cruelest irony in oncology. The single most important target in one of the deadliest common cancers was also, on a molecular level, one of the hardest targets anyone had ever tried to drug. This is Conference Hype. Stay with us, coverage continues.
Here's the biology problem. RAS proteins are molecular switches. GDP-bound is off, GTP-bound is on. The mutation breaks the off switch, so the protein gets stuck flipped on, constantly signaling the cell to grow. Sounds simple enough to block. It wasn't.
The problem is the protein's surface, a smooth, featureless ball with no deep pockets for a small molecule to wedge into, binding GTP with picomolar affinity, about as tightly as a molecule can hold onto anything. Early attempts like farnesyltransferase inhibitors failed outright. By the 2000s, most of the pharmaceutical industry had simply walked away from RAS.
Two people kept the field alive through that dead period. Frank McCormick at UCSF had been working on RAS since the mid-1980s. He helped discover the gap proteins that regulate the RAS switch, co-founded Onyx Pharmaceuticals, and spent decades arguing RAS wasn't undruggable, just not yet drugged.
And in 2013, Nobel laureate Harold Varmus, then director of the National Cancer Institute, used his position to do something the private sector wouldn't. He launched the NCI-RAS initiative at the Frederick National Laboratory, pooling public funding into shared reagents, structures, and assays so any lab could pick up where industry had given up.
This is Conference Hype. Stay with us, coverage continues.
The breakthrough came from an unexpected direction. Kevin Shawkat's lab at UCSF, encouraged by McCormick, screened roughly 500 candidate molecules and in 2013, published a discovery that upended the field's core assumption. They found covalent binders that could grab a cysteine unique to the G12C mutation, revealing a completely new pocket on the protein's inactive, GDP-bound state, the switch II pocket.
That mattered because everyone had assumed only the active, on state was worth targeting. Shawkat's work proved the off state had a weakness too, a discovery that directly led to the first approved KRAS drugs, sotorasib and adagrasib, years later.
G12C inhibitors were a huge win, but they only work for one specific mutation, and it's not the one that dominates pancreatic cancer. The next leap came from Harvard chemical biologist Gregory Verdine, known for pursuing what he called drugging the undruggable. At a company called Warp Drive Bio, founded around 2012, his team chased a much stranger idea: a molecular glue that could recruit a natural cellular protein, cyclophilin A, to clamp active RAS in place.
It was viewed as a long shot inside and outside the company, and early animal data raised real concerns about toxicity from blocking healthy, wild-type RAS along with the mutant kind. Revolution Medicines, founded in 2014, acquired Warp Drive Bio's program in 2018, and pushed the chemistry further, engineering it into an oral multiselective inhibitor able to lock mutant and wild-type KRAS, NRAS, and HRAS in their active on state, exactly the broad coverage pancreatic cancer needed.
This is Conference Hype. Stay with us, coverage continues.
That chemistry became daraxonrasib, RMC6236, in development, an oral, once-daily, non-covalent tricomplex inhibitor. On May 31, 2026, in a plenary session at the ASCO annual meeting, Brian Walpin presented RASolute 302, a global phase 3 trial of about 500 previously treated metastatic pancreatic cancer patients, more than 90% of them carrying RAS G12 mutations.
Numbers: median overall survival nearly doubled to 13.2 months with daraxonrasib versus 6.7 months with investigator's choice chemotherapy, a hazard ratio of 0.4, roughly a 60% reduction in the risk of death, with a p-value smaller than 1 in 10 billion. In the RAS G12 population, progression-free survival was 7.3 months versus 3.5 months. Response rate: 33.2% of patients saw their tumors shrink on daraxonrasib versus 11.8% on chemotherapy. Patients on the drug also reported better quality of life and slower decline in cancer-related pain.
When the survival curves went up on screen, the plenary hall reportedly gave the data a standing ovation, a rare reaction at a scientific meeting, in a disease that has beaten drug after drug for decades. The results published simultaneously in the New England Journal of Medicine, and the FDA has since granted expanded access and accepted the new drug application under an accelerated pathway. That's the RASolute 302 story and the 40-year road that got there. Tag us on X @WithYourTake and subscribe to Conference Hype for more evidence-first breakdowns of the biggest moments in oncology.